This is Will to Win, Overcoming Long COVID. I'm your host, Jillian Angeline, and my goal is to raise awareness about the hundreds of millions of people who are suffering months and in many cases years after contracting the COVID virus. I know about that myself. 6 surgeries, 5.5 years later. I'm on a road to recovery and healing, but it hasn't been easy. Now, of course, these episodes are for informational and educational purposes and do not constitute medical advice. Please connect with your medical professionals before trying anything we discuss during these episodes. Now, let's get started. Today's guest is Dr. Resia Pretorius, a distinguished research professor out of Stellenbosch University in South Africa. She's an expert on blood coagulation and circulating inflammatory markers in conditions like COVID. All right, Dr. Pretorius, thank you so much for talking with me. I really appreciate your help in chatting. You are one of the preeminent researchers on clotting, especially in the long COVID community. First of all, tell me what got you interested in the world of blood early on pre-COVID times.
Thank you for this wonderful opportunity to discuss our work with you. My research began a long time before acute COVID, probably around about 2003, 2004. I started working with blood and with fibrinogen and with clotting pathologies in a variety of diseases. I looked at diabetes, rheumatoid arthritis, psoriasis, Parkinson's, and Alzheimer's. And we've been studying blood and blood clotting since then. Around about 2011, I met up with my long-standing collaborator, Douglas Kell, because of our, we both worked on iron and our interest in iron dysregulation and how it affects blood and blood clotting.
Tell me about what this experience has been like for you over the past 5 years. I mean, for a lot of us long haulers, it's been quite an uphill battle. People have used that term for what you've been doing, speaking out constantly worldwide about the challenges that you've seen and what you've seen under microscopes. I remember early on, I wanted to literally fly to Stellenbosch University to get my blood looked at by you. I mean, that was something that was on my list early on. Of course, thank God for Dr. Vaughn and others stateside who could do something like that. But what has this experience been like for you, trying to help so many people?
It's been quite a challenging experience, but also a fantastic experience. In the past, before acute COVID or long COVID, our work was never so much focused on patients and patient experience and patient ideas on how to handle their disease and what they are feeling and how they are coping. COVID changed everything. Suddenly, we were exposed to patients asking desperate questions. They didn't know what's happening. 1 or 2 of our publications came out in 2020, 2021. And then it just exploded where patients contacted us, clinicians contacted us, researchers were interested. So it was a fantastic experience being closely involved with a fantastic research team over different continents that we have been collaborating with to try to look at the various pathologies, not only clotting and clotting-related or thrombotic endotheliitis, as we would like to call it, in long COVID. So it's been very difficult, but very rewarding and fantastic to meet all the patients.
I've seen that term endotheliitis mentioned quite a few times in research that I've done about you. Can you define that for someone who may not know what that is?
Absolutely. So if we look at our blood vessels contain the endothelial layers or the vascular layers. That is the cellular layer that covers the inside of all our blood vessels, from your tip of your toe to your brain, to the blood-brain barrier, is all vascular. So if we look at all the pathologies related to long COVID and acute COVID, all of the inflammatory molecules that are in circulation in these type of disease, or any post-viral disease for that matter, you will find all of the inflammatory molecules that are flowing through your blood vessels, attaching to your receptors on your vasculature, your endothelial layers, and it basically causes the whole of your endothelial layer from everywhere in your body, systemically. It causes these layers to become inflammatory. So you are systemically inflamed, and the term for that is endotheliitis, or inflammation of the vascular layers. And then we also associated it with thrombotic endotheliitis. So thrombotic means that associated with all of the circulating inflammatory molecules, all the damage that the endothelial layers have been exposed to by all the cytokines, the spike protein in circulation, also causes clotting and platelet abnormalities. And so therefore, we typically use the term thrombotic endotheliitis as one term suggesting what is one of the pathologies in long COVID.
I remember early on you would use the term sticky blood, and that's something that I've seen firsthand for myself, and a lot of people talk about. Even just in a recent post that you talked about on X, on formerly Twitter, you had said, we talked about sticky blood back in 2021. I wish doctors had paid attention then. What kind of progress has been made on the sticky blood theory since 2021 to now 2025?
So why we use this layman term sticky blood is we have found that all of the inflammatory molecules that are in circulation can attach to each other and on certain parts of immune cells and immune cell products like the neutrophil extracellular traps. All of these inflammatory molecules become sticky and they attach to each other to form these amyloid complexes, or as we call it, amyloid fibrinoid microclot complexes. So it's a lot of things entrapped. Now, our recent research have been using a very good program that was developed by a Polish researcher, and it's called Ameliogram. So Ameliogram is a program where you can put in the molecules that you think are of interest, and in our case, our molecules are of interest are the molecules that we have found with proteomics analysis that were entrapped, that we were found that were entrapped inside these microclots that we digested. So we have looked specifically at whatever is entrapped in these little complexes. You can take the sequence of these molecules. So I'm talking about von Willebrand factors, serum amyloid A, all of the inflammatory molecules that we and others have found in long COVID to play an important role. And you can put the sequence of the protein into this ameliogram, and then you can determine the amyloidogenicity of the molecule. And the amyloidogenicity in layman's term would mean how sticky these molecules are and how they are able to themselves drive amyloid protein misfolding and changes, and then when they are in circulation attached to each other to form these microclots that we are now calling fibrinoid microclot complexes, because there are a lot of complex interactions that they have. It's not a single molecule that forms these complexes. And in many cases, there are lots of cellular debris from the damaged endothelial cells, from the platelets, from red blood cells, nets that I talked about, that are entrapped in these entities. So the molecules become sticky, and the reason why we use the term sticky is because of the amyloidogenicity. And some patients, as you might have seen yourself, report that when they draw blood, the blood just can't flow out of their arteries in their veins, or it just clots right there in the tube. So that's why we think it's sticky, because of the amyloid propensity that we see in the blood samples that we get.
So this might be a layman's concept, but when you're looking at all of these inflammatory molecules under a microscope or something, I guess the hope is always, can you add something that will attract those molecules to pull them away from the cells and from those, from that chain? Is there a way to pull it off, or is that something you're still looking at?
We are looking at a variety of molecules, and myself and Dakel have already published on some of it, and many people are using some of the molecules that are known to break open and digest enzymes that can break open and digest these structures. And one of them is a product called nattokinase, which is the enzyme from natto, the Chinese product that people eat, natto from soybeans. And this is a very well known, long before we have looked at it, it is known to be clot busting, so enzymatic digestion. And then there's molecules like serapeptase, also lumbrokinase, which is also various enzymes. And then obviously in the past, we have looked at anticoagulants, and we have not used it in a trial, so not at all in any trial or in any such type of study, but we have been getting samples from various clinicians who have been using anticoagulants that prevent the clot from forming in their patient groups. And then we have looked before and after, and we have seen lots of good results from patients that have been using the traditional anticoagulants. But as always, these things are obviously need to be in a trial. They can be very dangerous. Anticoagulants can cause you to bleed and you can die. So we are exploring other types of molecules like nattokinase, serapeptase, and then other people like the Petrino lab. You might know that in Mount Sinai, they are actually looking at lumbrokinase currently in a clinical trial. And I'm very hopeful to have their results out very soon to see how their patients are doing in a setting where it's a perfectly looked after trial. It will be fantastic to see their results, to see if whatever's in circulation, which all the sticky complexes that we found, whether lumbrokinase can adequately break it down and have the patients also feeling better. That's really looking forward to those results.
Something that you're working on. It showed up in the news just a few weeks ago about these zombie cells, senescent cells, where the cells are kind of killed, but not really. And then they become immune to the immune system. And now they're just rolling around, creating inflammatory molecules or causing more inflammation in the body. Talk with me about that kind of research. How long did it take you to get to the point where now you're talking about the senescent cells?
It's work that my wonderful postdoc student Massimo Nunes is doing. So he's also the first author of the senescent cell experiments that we are planning, and the specific paper that you are talking about is currently under review, and we hope to have that accepted soon. And it's done. The actual paper was written with various co-authors: Bertram Fielding from Stellenbosch, Doug Kell as well, and then the Wust group. Rob Wust from University of Amsterdam is also included in there. So in that first dipping of our toes into the senescence environment is where we propose that during infection and during post-viral diseases, and with all the inflammatory molecules and viral molecules and inflammagens in circulation, that our endothelial cells, so the layers of the vessels, once again, and it brings it back to our thrombotic endotheliitis, they go into senescence due to the onslaught of constant inflammatory molecules attaching to the receptors, and then it drives them into sleep mode so that they don't really function as they should. And that will cause all sorts of issues with oxidative stress and not being able to transport oxygen and products over these layers, and that is a real problem for us. So the next experiments, together with one of my other postdocs, Claudia Christovitz and Bertram Fielding and Massimo Nunes also, we are looking at the roles of microclots, these complexes, the fibrinolytic microclot complexes, on how they can interact with endothelial cells. And we hope to have some data from the laboratory very soon, in the next few months, to actually show what happens when these complexes are exposed to cell lines, which is endothelial cell lines that's in our laboratory, in our labs, and how we can find out how these molecules entrapped inside the microclots and in the plasma of these individuals interact with endothelial cells, and perhaps give more insight on how it will drive these cells into senescence.
Thank you so much. I know that the article I'd read talking with you all mentioned a registered clinical trial here in the US, hoping to do some research about these zombie cells. So I think that that is the goal ultimately, to see how those zombie cells can go away in those clinical trials. Because I think clinical trials, from a researcher standpoint like yourself, clinical trials are the key to be able to getting more data, correct?
Absolutely. But our focus in our laboratory is not to be involved in clinical trials. We are the laboratory researchers who find new diagnostic markers, who develop new methods to be used in these trials. So we are sort of I think, the basis on which trials might then build, to use a
Mhm.
lot of other things that they would typically use in trials, and to analyze cytokine levels and proteomics analysis and
Mhm.
markers of how the patients feel, and whatever you would typically do in a trial. But we are building new methods, new diagnostic tools, using new or novel methodologies like the imaging flow cytometer, or all sorts of other types of instrumentation that might be adapted to see the unseen. So looking at things that nobody else has looked at a specific, in a novel way. So that's our focus, to provide new insights with new experiments, new biomarkers, new methods.
These labs in Stellenbosch that you are using, have you had to add new equipment or add anything as you've changed course from the studies that you were doing pre-COVID to now?
We actually did, but the road to this was difficult, but absolutely rewarding. And I must say, in 2021, I partnered with Kernels, which is a crowdfunding institute, together with PolyBio Research Foundation, and then Balvi Research Foundation came on board. And Amy Prohl is just absolutely fantastic from PolyBio. She and Balvi Research Foundation and Kernels allowed us to crowdfund for an imaging flow cytometer. And we would definitely not have had it if it wasn't for hundreds, thousands of patients who assisted us with small donations, big donations, and we could
Right.
crowdfund for this imaging flow cytometer. Within 6 months after we started, we had the instrument in our lab. So I'm always, actually, I can't even think how fast it happened. I'm still, I can't believe it. So I was so happy to have that. And if it wasn't for that, we would not have had a way to get the data
Mhm.
on a large scale. And because in the past we used microscopes, and
Mhm.
one question that we always get is, oh, so well, you can cherry pick, you're under a microscope, you look at this, you can take photos randomly and then you say something. With an
Mhm.
imaging flow cytometer, you can't do that. You
Mhm.
put in your sample, you run it, you've got your gate set up, and the data is what the data is. The numbers and the sizes of the distribution of cells and of, we developed actually a novel method on this imaging flow cytometer to characterize and quantify these amyloid microclot complexes. It was never done before. If you look at a flow cytometer, it's a cytometer for cells, and we adapted the method very fast. So if it wasn't for patients, clinicians, family members of patients, and these wonderful foundations, we would not have had these instruments. So, and Balvi has constantly supported us together with PolyBio. If we ask, they give, and it's fantastic.
Mhm.
Balvi has assisted us with some other instrumentation, automated microscope method to look at platelets. um, Balvi and the sister association and foundation, Kanru, is now currently funding Massimo, my postdoc that I mentioned previously, for a 3-year postdoc in my lab. If it wasn't for those philanthropic
Wow.
fundings that we just get from random people, we would not have been able to do this research as fast as we have.
I mean, and talk about that kind of collaboration with the patients, with the funding, but also with someone like Professor Kell in the UK. You're working with Dr. Jordan Vaughn here in the US. What is that experience like to collaborate worldwide? Because something like COVID didn't know any borders, didn't know any boundaries.
People like Dr. Vaughan, who is a clinician, is not a traditional researcher, but he read our research papers very early on in the pandemic, and he saw what we were saying in his patients. And it's clinicians like him that have come to the fore. I've got so many of such similar type clinicians who took the time, read our papers, however basic research it was, and translated it to actually make a difference in their patients. And clinicians like Jordan Vaughan is, it's just absolutely fantastic to work with him. They have got a brilliant scientific mind, but also the clinical mind associated with it. And then obviously Doug Kell from Liverpool, we've been collaborating for forever. It seems I know him forever. But we have set up so many more collaborations with Harvard, with MIT, with Mount Sinai, Yale, Derby University. We've got a fantastic collaboration with Mark Fahey. There are so many. Ray Duncan, Benita Kane, clinicians in the UK. Many more, including Innes Laboratories, who's a startup company in Switzerland. We've patented our method to look at
Wow.
microclotting and imaging flow cytometry, our methods. And we're doing it through our startup company, Bioco Technologies, who is doing a fantastic job at the back to try to optimize various methodologies. Innes Laboratories have partnered with Biocode, and they're doing the imaging flow cytometry test in their laboratory for patients. So patients all over Europe are sending their samples to them. It's been an absolute fantastic experience to collaborate with all of these individuals who are just looking for new methods and for new ways to assist patients. And then we've also been working with Professor Alain Theory, who is a netosis expert from INSERM in France.
Wow.
So many people to mention, and I'm obviously leaving out a lot, but those are just the many of the researchers we are currently collaborating with.
So what do you need next? It sounds like you've had, great connection, you've had funding from different sources. What is your greatest need at the moment? Where's something that you say, if you leave the lab one day and you go, I wish I had X, or I wish I had a little bit more time to research on this. Like, what is your next battle for you right now?
I think the battle for us always is funding. It's really we've got the samples, we've got material and data sharing agreements from all over the world to get samples from well characterized sample groups. I think the most important thing for us is philanthropic funding. We've got a lab fund where people donate grants into or funds into. So we always try to do proteomics. That is very expensive. So funding for proteomics and more equipment is always obviously what our goal aim is to get. Obviously, we are applying for lots of grants as well, but grants take time. It can take a few years for it to actually realize from application to actually getting it in different rounds. So philanthropic funds and grants have been our lifesaver, absolutely, and we've been just so thankful for all of those. But we need more.
To simplify something, can you define proteomics for me?
Okay, so proteomics is a quite a specialized type of technique where you take proteins, any proteins, you digest it. And that's what we have been doing in 2021. We started with that also. We have digested our microclots, our amyloid microclots. We broke it up so that you can literally tease out all the proteins, and then you run it through a specialized instrument, a mass spectrometer instrument. We don't have one in our lab, but we send our samples away to our Medical Research Council. that have got the equipment, and we've got a fantastic biochemist, Dr. Marie Vloek, that is our associate. He was at Stellenbosch University previously. He's now doing it on his own terms. Then you can understand and find out what are the actual molecules, the proteins, or the enzymes, or the antibodies, because antibodies and all sorts of antibody-related molecules, cytokines, are all proteins. Then you can understand what the concentrations are, and you compare it with a healthy individual, you compare it with diabetes or other groups, so that you can understand what are the inflammatory molecules that are in circulation and might drive the disease progression. So proteomics, quite a specialized method, quite expensive. We were very lucky to have funding from Polybio Research Foundation, and many of us, and standing up to POTS is one of our other philanthropic organizations that have supported us fantastically. We've just completed a proteomics analysis for with POTS and long COVID with Professor Satish Raj's group from Canada, and we are teasing out the molecules. So proteomics is basically a way to study the content of the protein which drives the disease processes.
Fascinating. Well, in my interviews with Dr. McMillan and with Professor Kell, they have said that it's so important because this kind of research about COVID has really brought to light the concerns about people suffering from ME/CFS, from Parkinson's, like you were doing that research ahead of time. So it's really put to the forefront with 400 million people at least suffering with long COVID worldwide. All of this research and all of the resources that are being pulled to do this kind of research will actually also help people that have been suffering for decades, where a lot of doctors have told some of those people with ME/CFS and others, it's just in your head. And I love one of the phrases I saw recently was, it's not in your head, it's in your blood. So a lot of this research, I feel like, will not just help long haulers, get back to square one, get back to where they were pre-COVID baseline. That's the hope, at least in my mind, for myself and so many others. But it's going to help, people who may have been suffering for years and years before all of this.
Absolutely. So ME/CFS patients are close to my heart as well. They've been suffering so much for years, and they have been belittled and gaslighted, and just to be told to just get out of bed and exercise or smell the roses, and they simply can't. They have been really, and it's a constant battle to try to convince researchers and clinicians that these patients are extremely ill. And it's so sad for me to look at X feed and still see the old research community who have been driving these gaslighting, and it's all in your head, just wake up and smell the roses type of people. It's so sad for me to still see they are continuing after many, not only us, but many other people have shown utility for methodologies that show that it's not just a psychological condition, that there's a real biological reason why you are feeling the way you are feeling. So it is fantastic for me to spend time to also interact with patients with ME/CFS, and to also look at their results and to find similar results in those patients as well. One of the important things that I'm very worried about is that Parkinson's and Alzheimer's disease have, traditionally been seen as of two conditions that are, only later in your life you have to start thinking about, in your 60s, 70s, 80s, you might start worrying about that. But I'm very worried that many of the long COVID patients might have similar experiences with perhaps developing these type of conditions earlier. So we need to urgently look at the links and draw the lines between these type of diseases to understand what is happening in Parkinson's, in Alzheimer's pre-COVID, what is happening in long COVID, in post-viral diseases, draw the links between them, find the similarities and where they're not similar, and then try to find panels to understand what you can test for, to identify these molecules early on, firstly, and then secondly also then to find possible ways of treatment.
Yeah, the therapies. That's what we all want to hear. How do we prevent something
Absolutely.
like this from happening? And I think that that's the kind of research, you're all doing to be able to inform those therapies. You're so far on the front lines and ahead of the game with a lot of this research, but you also need that follow through from so many others, which it sounds like you have a huge support system, which is great to hear. And it sounds like even though you feel like you may be shouting from the rooftops on a lot of this content, do you feel like it sounds like you're being heard?
I think we are being heard. We've been in contact. Our lab and our collaborators have been in contact with numerous clinicians, patients, and big organizations as well. The NIH, WHO, they know about us. They know about our work. We've been talking, I think, collaborators like Dr. Vaughan has been talking at Senate in the US. He's been in contact with various of the people that make the decisions on how to handle and treat patients. So it's not only us. I think all our collaborators are doing a fantastic work to talk and to talk to associations, cardiology associations, hematology associations, to get the word out there. So it's not only us doing these things, it's the broader long COVID research community is doing a fantastic advocacy work to try to make people aware. And it's going to take time because
Mm,
people are set in their ways. Clinicians have learned stuff in textbooks. This is a new disease with no textbooks. We need to write them. So currently, if you talk about post-viral disease in medical training environments, it's not really adequately studied by clinicians in training. So we
mhm,
need to provide new insights and try to convince them to look at research for their clinical practice. But it's difficult because obviously many of these young clinicians are trained by older clinicians that have a certain insight. That's what they were taught. That's how they do it. Uh, it's going to take long to change insights and change the rhetoric, but we will need to do that. Otherwise, we're going to lose patients to be bedridden and not be able to participate in any of the world economy. And that is so sad. Patients that don't see their children grow up, that won't be able to see how their children graduate or go into life with them to celebrate. It is really terrible to just think about that. So we can't think about that. We need to
mhm.
find solutions, and that's what we're trying to do.
And that's what I love to hear. I feel like you've been so solution oriented the past however many years that you've been working this, but the 4 years that I've heard about and known about you, I can tell that you keep fighting that good fight, and that you're one of the people that's going to help make those changes. And it's great to see that you're continuing to battle that. Considering this is the Will to Win podcast, it I ask a lot of the people that I interview, do you have hope that things can change, that patients like myself and so many others can get back to where they were? And I feel like you're going to say yes, but I just want to give you the opportunity to share your
I
thoughts.
don't have hope. I have knowledge. I think we will succeed. I'm not hoping that we will succeed. We cannot even hope now. We just need to get solutions. I think we've got the tools, we've got the people, we've got the will to make it happen. The trials are happening, funds are available, how difficult ever, but we've got people that fund. We've got some of the associations
Mhm,
welcome and all of those, the MRC for different countries, the NIH, some of the European councils that provide funding. I just hope that they can filter down faster, but
mhm,
I really feel that we will do something, and we will change the way that we see long COVID patients. We will continue, and because we don't have that. We can't give up. Uh, we can't. If we fail, then we fail millions of people, and that's not a possibility. We cannot fail, unfortunately. We need to do this.
I agree 100%. And it's so good to hear you say that. If anyone wants to see more about your research, how can they find out more, Dr. Pretorius?
So I am on LinkedIn, and I'm on X so it's at Resia Pretorius. We regularly post all our preprints and our publications. We will be posting a next preprint within the next days. Also, some of my students work, Massimo Nunes, the postdoc that I've mentioned previously, first also on pseudoserum. So they can follow me there, and I always post all our preprints and our finally published papers and some comments as well. So that's the easiest way to find me.
And from someone who's not directly in the medical community, when you say preprint, what does that mean versus something that's more peer reviewed? How does that work?
So the past few years, there have been a certain move away from the traditional peer-reviewed process by adding an additional layer to it. So if
Mm.
you've written your paper and you are happy with the standard of the paper, then you preprint it to a preprint server where it's got a dedicated DOI. So if it's made, if you preprint it, you can't remove it. It's there forever and ever. So you've got a DOI link to it. People can comment and read it, and then the researchers, us as researchers, then take the publication and we submit it to a journal like Cell or Nature or some of the other cardiovascular diabetology or whatever journal that we are interested to submit it to. It will then go into peer review. And eventually, when it's accepted, it will be linked back to the original preprint so that it becomes one document, so that someone reading the paper can see the preprint and how the changes or the paper evolved after various peer review stages. So it sort of involves other researchers and clinicians and readers and patients in your research before it's actually published. Many of the journals, the large journals, the big gun journals, they are also providing a link when you submit your paper. If you haven't preprinted it, you submit your paper to Lancet or to one of those journals. They've got a specific link to say, do you also want to preprint? So then the paper is sort of in review, but also available already to see your data. What makes that quite nice is if I typically would have submitted a paper to a journal, it might take anything from, if you're really lucky, 6 months, but usually 8 to 12 months before the paper is through peer review, actually published and then visible for for anyone to read. But now in the meantime, 12 months or even six weeks can be too long in our environment. Then the data is already old in your lab and you've now already moved on. So now it allows the patients, clinicians, and researchers to interact with your data before you actually publish it and perhaps make comments. And in some instances, some of the authors would then also look at the comments and say, oh, perhaps I should have done that experiment. And you can even upload various versions of your
Oh.
preprint. It's just in version 2, and you see, as you go about. So it's peer review by your peers, by clinicians, by researchers before it's actually accepted. It's really a fantastic opportunity for researchers, and we always preprint our work long before it's actually finally been accepted. Obviously, it's the beta version of the paper, and many things change. And then some researchers, have an experience, and we ourselves have had an experience that you preprint something and you simply can't get it published for some reason. But then the preprint at least is there, and then you can change your methods or look again at what you are doing and pivot to another method. So it really is really helpful for researchers and for clinicians to read such preprints.
I also think it's validating for patients too, because for a lot of patients, when I saw a preprint about post exertional malaise and exercise, induced issues tied with long COVID. It felt so validating to know that, oh, this might be why I struggle sometimes if I work out too hard. So even if a preprint is out there, it may take 8 to 12 months to officially be published, but it gives that validation and that hope that, hey, maybe there will be therapies, or it will inform future, physical therapy options for someone like myself. So I think it just provides that extra layer of hope, like you're talking, and it provides not just hope, but knowledge for a lot of us out there. Like you were saying, knowledge is power, especially in this world that you're in. Well, I want to thank you so much for talking with me and for taking valuable time out of your busy day to chat. So if you have any other questions, feel free to reach out to me. And thank you so much.
Thank you. It was so nice talking to you. And I really like such platforms because it assists us to also talk about our research and our vision for what we are doing.
If you have any topics or suggestions, I'd love to hear them. Contact me on Instagram, Angeline_travels, or on X at JL Angeline. I'll see you later.